So we're going to start having a look at the digestive system today and what we call an accessory organ for the digestive system. So the digestive system consists of the digestive tract, which you might have heard called the alimentary canal. So from the mouth to the anus. And then there are other organs which are called accessory organs, and the liver is one of them. Another accessory organ is the pancreas and the gallbladder. So all of these things are secreting substances into the digestive system. So not necessarily part of the main tube, but secreting substances in. So we're going to have a look at the digestive system, but also part of this process involves specialized proteins called enzymes. They're a really fundamental part of the digestive process, breaking stuff down. So we'll also be looking at these molecules as well, these proteins. So understanding the digestive system then, let me just move something on my screen so I can see what I'm doing a little bit better. Okay, so we're going to start having a look here at the sources and functions of dietary components. So essentially this is our food. We need to eat carbohydrates, which we get from sources such as pasta, sugar, rice, potatoes. Lots of roots are full of carbohydrates because plants store their carbohydrates as starch. So if we eat grains, you know, the plant seed, they're full of starch. If we eat the plant tubers, the roots like potatoes, they're full of starch. So we eat the plant carbohydrate store starch and extract energy from it. We also need to eat proteins. So good sources of proteins are meat, fish, mushrooms, beans. We need proteins to break them down into their subunits so that we can rebuild them as different proteins and to allow us to grow. Remember, we were looking at those structures within our cells not so long ago. A lot of those structures that we looked at last week have got protein components. So we need proteins for growth. We also need to eat lipids. So that's the scientific term for fats and oils. The difference between fats and oils is literally just whether they are liquid or solid. So oils are liquid at room temperature and fats are solid at room temperature, so around about 22 degrees. So oils would be things like olive oil, rapeseed oil, nut oils, that sort of stuff. And fats that you might come across would be animal fats, so things like lard or butter are fats at room temperature. Fats are really important for providing energy, like our carbohydrates. We can break them down to produce energy. They can go into something called the Krebs cycle if we break them down. We'll learn about that much later on. It's how we get our energy released from usually glucose. But they're also very important. Remember the structures we looked at last week? We looked at that membrane structure, the one that was like the lava lamp. All of those membranes are made up of a fat or lipid, really, substance. So lipids are really important for forming the membranes around our cells and also within our cells, the structures within our cells. We also need to eat fiber. So fiber is actually the cellulose cell wall of plants. So we eat a lot of plants, or at least we should. And the cellulose cell wall, we can't break down. We don't have any enzymes, any molecules that are capable of breaking these cell walls down. So that fiber literally just passes straight through. It's digested in our feces. But it's really important because it's what pushes our food along the digestive system. If we didn't have fiber, our muscles would have nothing to sort of push onto to push our food through. So that fiber is a really important component, even though we don't actually extract anything from it. We don't digest it and break it down and take the molecules into our blood. It is just there to bulk up our food and help push it through the digestive system. Then we've got a whole load of vitamins. I've only selected to look at. We have vitamin A, vitamin C, and vitamin D. Sources of vitamin A are liver. We can get vitamin C from fruit, and vitamin D from egg, but also sunshine is the main source of vitamin D. And these are important in preventing disease, so nutritional disease. So vitamin A is important for our vision, and it helps blindness. Vitamin C prevents scurvy, which is like a bleeding and vitamin D prevents rickets, and it's needed for calcium absorption as well. Then we have some minerals here. So calcium is a mineral and iron is a mineral. Calcium we can get from milk, and it's important for healthy bones and teeth, and iron available particularly in red meat. And we need the iron to make the hemoglobin molecule, which is in our red blood cells. We also need to consume a certain amount of water. So that's available in lots of food, particularly fruit, which is very water-based. And obviously we can drink it as well. Now we need the water because most of our bodily processes, our reactions take place in an aqueous environment. So they're water-based reactions. So we need it for almost every biochemical reaction in our body. We also lose a lot of water through respiration. When we breathe out, we're breathing out water. It's something that's produced in our body, so it gets excreted in our breath. We also lose it through urine, and we use it through sweating as well, which is part of the cooling process for our skin. So those are our main dietary components then that we should be consuming on a daily to weekly basis. When we talk about the digestive system, it's not just a process of digestion. Digestion is just part of the process. So to gain or take on nutrition, we need to ingest the food, which is basically eat it, digest it, which is to break it down. We need to absorb it, which is to take it into our blood. We need to assimilate it, which is build it into our cells, and then we ingest the waste. So note, we don't excrete the waste, we ingest the waste. So we're going to look at these processes then. So we'll start at the top end and follow the passage of food through the system is probably the easiest way to do it. When we eat, we ingest the food through the mouth. The proper term for the mouth is actually the oral cavity or the buccal cavity. The mouth itself is actually just the opening. The inside where your teeth and your tongue are, the correct term really is the buccal cavity or the oral cavity. So the food enters through the mouth, which is the opening, and then we chew it up. And when we start to chew, we're breaking it up physically or mechanically, if you prefer. So we're physically breaking it into smaller pieces, and we're also breaking it up chemically, which we'll see in a minute. So this physical or mechanical digestion, because it is you're breaking something down, so you are digesting it. This physical or mechanical digestion from chewing, first of all, increases the surface area of the food. That's really important because there's a bigger surface available for all of the enzymes to work on that we're going to talk about in a minute. And chewing the food and breaking it up physically with our teeth and our tongue, actually, the tongue's a very powerful muscle. So chewing and breaking up that food also mixes it with saliva. So that's important because it helps to lubricate the food. And then once the food is lubricated, we call this food now a bolus of food. It's just a lump of food mixed with saliva. So it's much easier to swallow the bolus of food because it's lubricated. So if you imagine eating something really dry, where you hadn't mixed it well enough with saliva and then trying to swallow it, it's actually quite hard. It gets stuck in your esophagus or stuck in your throat. It's not very easy to swallow at all if you haven't chewed your food properly and you're eating something really dry, like crackers or something. Is a good. I wouldn't suggest try this, but you might have had experience doing something like that, or eating a load of bread with, it's quite hard to swallow. So The tongue's important in this process. It helps to mix your food with your saliva. You're increasing the surface area by breaking it down with this physical, mechanical digestion, and you're lubricating it. So we've increased the surface area, we've mixed the food with saliva, and that saliva also contains an enzyme. So if you've studied this at school, you might remember this enzyme. It's the one that people tend to remember if they're going to remember one at all. So the saliva contains an enzyme called salivary amylase. So salivary just means it's in the saliva, and amylase is a carbohydrate. So that means it breaks down carbohydrates. And this amylase particularly digests starch. So as I was just saying a moment ago, we eat plant materials, and plants store their carbohydrates as starch. So things like rice, potatoes, there's loads of starch in them, which is great for us because if you look at this, this is a big long chain of glucose molecules. So starch is made up of glucose, and glucose is actually what we need for our cells. It's where our cells get their energy from. So starch is a great source of carbohydrate energy for us, which can go straight into this energy reaction, respiration, that we're going to learn about later on in the course. So by eating starchy food and breaking it down, we get access to loads of glucose, which we can use for energy. So the first enzyme that we meet in the digestive system is salivary amylase. And salivary amylase breaks down this carbohydrate. Starch is a carbohydrate. It breaks down this carbohydrate into smaller chunks. So it breaks it down into 2 glucose molecules together. So much smaller chunks. They're only 2 glucose molecules big. That's called a disaccharide. So saccharide means sugar. So, you know, you've heard the word saccharin, sweet. It's just that's what it means. Disaccharide means 2 sugars. So salivary amylase breaks down starch into this disaccharide, this 2 glucose molecule. And that happens in the oral cavity. That's the first bit of chemical digestion that takes place. So we haven't even swallowed our food yet, and we've already begun the process of chemical digestion. Starch is broken down by salivary amylase into that disaccharide, those 2 glucose molecules. It's actually called maltose, that disaccharide. So it's broken down into that disaccharide maltose, and we have the physical digestion, the chewing, the increase the surface area, and the mixing with the saliva. So what's happening in this digestive process that we've already started in the oral cavity is we're breaking down large molecules into small soluble molecules. So starch is a really large molecule. It's made up of lots of glucose molecules stuck together, but it's not soluble, which is a problem because remember, it's taking place in a watery environment in our cells, and not small. So this molecule is big. You can see that it's a big chain, smaller and soluble. And those glucose units, you can see our starch molecule is made up of, they're small and they're soluble. So if we can break down this big chain of glucose into individual glucose units, insoluble molecule to small soluble molecules. It's still a little bit too big at the minute because it's 2 glucose molecules, a bit smaller still. But we're breaking down these large molecules into smaller molecules. So, for example, our carbohydrates we eat tend to be starchy. Starch molecules into the smaller glucose molecules. And similarly, we need to break down lipids into their subunits, we call them, out of, and we need to break down proteins that we eat into their subunits as well and absorb them. So the large molecules are broken down into small molecules, absorb through the wall of the gut and get them into our bloodstream. Once bloodstream, particularly these glucose molecules, we can transport them all around the body to where they're needed. For glucose, we need inspiration, we need them for energy. So it's particularly important that we can get them digested and transported around the body. Right, so we've chewed up our food. We have a bolus of food, like a lump of food, mixed with saliva, lubricated to help us swallow it. So we swallow it and it goes into our big tube that goes down to our stomach. There's a little flap which stops us inhaling our food most of the time. It's not particularly efficient. I'm sure we've at some point or another eaten food, particularly when we're younger. It tends to happen occasionally. So it's not a design. If we were designed rather than evolved, you would not put the and the swallowing apparatus in the same place. So some people say good evidence for evolution rather than design by a creator, for example. But anyway, that's evolution is another unit. So what happens when we swallow is our food enters our esophagus, which is a tube, and it's mostly made up of smooth muscle. Talking about muscle tissue, weren't we? Smooth, skeletal or striated cardiac muscle last week. So this is an example of smooth muscle. There are two layers of the esophagus. There's one going down, which is called longitudinal because it goes down the length, it's long. And then there's circular that goes around the tube. So, you know, not very imaginative in the names, but that's good because we all know what that means. We've got longitudinal, we've got circular going around in a circle. So the food doesn't swallowing into the stomach by gravity. If you stood on your head, food make its way to your stomach, it gets pushed there by alternate contractions of these two muscle groups, well, contractions and relaxations. So the food bolus, the food lump, which is lubricated, what happens? Muscle above it will contract, so essentially squeezing tight. And while that's happening, the longitudinal muscle below will relax. Circular is tight and the longitudinal will open up, and that will squeeze it down a little bit, and then contraction will move down a bit. Got circular muscle contracting above it, longitudinal relaxing below it, and so on until the food pushes down from the mouth down to the stomach. Okay, let's have a little look at some questions and see what's gone in so far. So you can write on the chat if you want to. I'll just grab the chat up so I can see if anyone's writing. And if you're not sure and you don't want to, you can write on some rough paper. That's fine too. Okay, I'll pop the answers up for you then. So first process required for nutrition is ingestion. So we go ingestion, digestion, and then absorption. The enzyme which we find in saliva is called and what's the role of the dietary fiber in the peristalsis? Well, it helps push against the fiber, the bolus, and it also absorbs water. in the digestive system further on in the large intestine. Okay, so once we have swallowed our food and it has been pushed by this alternate contraction of the circular and longitudinal muscles or peristalsis, it finally reaches the stomach. Now, the stomach is a muscular sac, and it also has circular and longitudinal muscles. You can see them here. Look, here's the circular muscles going around the stomach in a circle. So around it like that. And you've got the longitudinal muscles going lengthways down the stomach there. So it's got those same two layers, but there's also an additional layer going diagonal, the oblique layer. So we've got the circular, the longitudinal, and the oblique layer. It looks really wrinkly on the inside here. We call the guy. And what they do is they allow the stomach to expand and fill up with food. It can expand massively. Your stomach can expand to hold up to 4 or 5 litres of food, which is huge. That's like a washing up bowl. So obviously that's not something you'd want to be aiming for most days, but that's how far it will expand in an adult male. Oh, an important structure of the stomach that we talked about, specialized epithelial cells, is the lining, and it contains holes called gastric pits. You can see them there. What's inside these gastric pits is special cells which release hydrochloric acid and pepsin. So pepsin is another enzyme. We already had salivary amylase in the oral cavity that breaks down. Now we meet our second enzyme, and that's pepsin, which, so protease, clues in the name, it breaks down proteins. The adaptations we talked about last week of this, the cells in this area, and that produces a mucus layer. The epithelial cells, which is really important because it protects these cells from harsh chemicals that are being released. So hydrochloric acid has a pH of incredibly acidic, really, really dangerous if it comes into contact with this. And pepsin, as we just said, is an enzyme, and it breaks down. If it came into contact with the stomach, tissue and proteins, it would break them down too. So we need to protect all of this epithelial tissue by secreting mucus around this area. So ingestion enzyme called pepsin and hydrochloric acid. So what's going on then in this area? Digestion of proteins. So the first bit of digestion of protein broken down here. It's a long process. They're big molecules, but they begin to get broken down here. So we're going to have a look at the structure of a protein. So you can have a look here. So this is globin protein. It's the one that's found in blood. So it's what we call a globular protein. It means it's in a blob type shape. And this is just a gel. Good one to use. So what this protein is made up out of is 4 subunits, and each subunit looks like this. It's a chain of amino acids. So that's the subunit that makes up a protein, a chain of amino acids. We'll look at these when we look at DNA and how proteins are built. But for now, it's just a chemicals, and they fold up in either bag or a helix. So a twirly, a bit like the old-fashioned phone. So you get your amino acids, they twirl up or zigzag, and then they form themselves to give us a subunit. And we've got happen to have 4 in this. And you can have 2, you can have 5, you can have 8. This particular one that's carries the oxygen in blood, this one has got 4 subunits. So that are getting broken down in the stomach by protease enzymes. So what is happening is these big globular proteins are getting broken down into shorter chain polypeptides. So they're getting broken down, just essentially snipped. This long chain and clumped together like this is getting just snipped by these enzymes into shorter chains. Let me just check. Oh, right. Apologies. The audio cut off for a second there. Yeah. This is a polypeptide chain. It's made of amino acids. So it's amino acids. They then fold either in a zigzag or in a sort of twirly helix. That's just because of the bonds between the molecules. But now we will cover it again later. What's important for now is the subunits of proteins are amino acids, and they're found in a chain which basically folds and up like this. So what's happening in the stomach is these clumpy proteins are broken down into shorter chains, So far we've had starch, carbohydrates broken down in the oral cavity into shorter disaccharides, so 2 glucose molecules, and we've got these big long chain in protein molecules just broken down into shorter chains in there. So there's no significant digestion of fats or carbohydrates going on in the stomach. In fact, we have had no digestion of fats or. At this point, we have a mixture in the partially digested food. So there's been some carbohydrate digestion, and it has been mixed with saliva that when we swallowed the bolus, and gastric from the stomach, gastric pits. So the hydrochloric acid. And this is now called chyme, this mixture that's in the stomach. I know it looks like it's pronounced chyme, but it's not, it's chyme. So this mixture is now called chyme, and it is essentially what you throw up if you vomit. It is not a very pleasant mixture. It's acidic and not very pleasant. You can get heartburn if that moves back up into your esophagus. It's not a very nice feeling. So it's a pH of around about 1 or 2, and that's important because what that kills most of the bacteria that you might eat with your food. Not all of it. With food poisoning via eating, but it will kill off a lot of bad stuff that we ingest. It will also, this pH, this low pH, this denature most proteins and activate pepsin. So we need this low pH to get this enzyme working, for this protease to start breaking down the proteins and the protein. We need a low pH. But also having a low pH, it disrupts these structures a bit as well. pH helps us to break down the proteins in our food, because this protein, which we see like this in its globular form, all wound up nice and neat, this pH, that low pH will start to interfere with the bonds that are holding this particular shape. So it will become unraveled. It will, and it will make it easier for this pepsin enzyme it will make it easier for that pepsin enzyme to start to do its job, because that protein said it's denatured. That means it's lost its shape. So it's in the stomach down there at 1 or 2. Okay, let's try a couple more questions. Enzyme in saliva begins the digestion of starch in the mouth. What's the primary role of chewing in the digestive process, and which enzyme is secreted by the stomach to digest proteins? Don't forget to have a go in rough if you don't want to write in the chat as well. It does help you process the information. Okay, I'll just pop the answers up. So the enzyme that breaks down starch in the mouth is salivary amylase. Remember, it's breaking it down into these disaccharide or 2-glucose units. The primary role of chewing is to increase the surface area of the food. It also mixes it with that enzymes and lubricates it. And the enzyme which is secreted by the stomach to digest proteins is pepsin, which is a protease. Okay, a couple more questions. What is the role of the hydrochloric acid in the stomach? What's the primary role of the tongue in the digestive process? And which enzyme breaks down proteins into the polypeptide units? Right, I'll pop the answers up for you. So the function of hydrochloric acid in the stomach is to activate pepsin. It also will break down or denature some of the proteins to a certain extent, and helps to protect us from bacteria that we might consume. The primary role of the tongue is to mix the food with the saliva, and the enzyme that breaks down the proteins is pepsin in the stomach. Okay, so one thing that is important with enzymes is temperature. They have an optimum temperature. So in humans, they're nearly always 37 degrees because that's around our body temperature. Some of them act a bit lower. So there are some involved in sperm production in the testes that prefer a temperature a little bit lower, like 36, 35. And that's okay because the testes are a bit cooler. They hang outside the main body. But pH is quite variable. The skin has a pH of about 5.5. The stomach is around 1 to 2. The digestive system can be quite alkali further on in the small intestine. So although some of the conditions that enzymes need are pretty stable temperature in humans, some of them are quite variable pH. So pepsin from the stomach likes a low pH to work best. So so far we had some preliminary digestion of protein. So the first bit of protein digestion that happens in the stomach. And then we need to have the terminal digestion, the end of the digestion of the, not proteins anymore, but they're polypeptides because they've already been broken down. So the polypeptides are broken down further into the individual amino acids in the small intestine. Also, if you remember earlier, we were talking about the starch in the buccal cavity or the oral cavity being broken down to these disaccharide units, 2 sugars big. These need to be broken down further. This is called maltose, by the way. These need to be broken down further into glucose so we can actually absorb the glucose. So, where is this final stage of digestion happening? What enzyme is released by the pancreas to further digest the starch, do you think? Have a look at those names and see which one you think is likely. And which enzyme in the small intestine do you think breaks down maltose into glucose? So have a look at those names and see if you can work it out. If you remember back last week, I said to you, anything that ends in A's is usually an enzyme. And you can usually work out quite well what it is working on based on its name. So see if you can work it out. Don't overthink it. Just have a guess and you'll probably be right. Okay, let's pop the answers up. So, final stage of protein digestion, the terminal stage or terminal digestion happens in the small intestine. And what is being released to digest that starch? Remember, it was called maltose now because we've got 2 sugar units. So the enzyme that actually breaks down the maltose into single glucose units is called maltase. And that's the answer for that one there. It's just phrased slightly differently. The enzyme in the small intestine breaks down maltose into glucose is called maltase. So terminal digestion and the rest of digestion or the end bit of digestion is happening in the small intestine. The polypeptides, so those amino acid chains, are broken down into actual amino acids. So just completely chopping up that chain. And the maltose that we've got from the mouth, the oral cavity, is broken down further using an enzyme called maltase into glucose subunits. So this glucose, the subunits of the carbohydrate, and the polypeptides, which are broken down into amino acids, the subunits there, the amino acids, can all be absorbed into the blood because they're small and can pass across the small intestine into the blood. So the small intestine then is this really wiggly bit, and it's wiggly because it is really, really long. So that's an adaptation for its function. It's long because that slows down the transit of the food through the system, and that slow movement allows all the nutrients to be absorbed. So the small intestine is broken up into 3 parts. We call them the duodenum, the jejunum, and the ileum. Small amounts of chyme are released from the stomach at a time. So we don't just go, right, mix up all the food in the stomach, mix it with the protease enzymes, and then whoosh, it all goes into the small intestine. There's a ring of muscle at the end of the stomach, and it really slowly releases food into the small intestine. It doesn't just open up and it all goes whoosh through. Little bits are released at a time. And that's important. You can just about see here the pancreas. It looks a bit like a feather. That is releasing substances. Loads of these enzymes that we were just talking about are released into the small intestine from the pancreas. And then also substances from the liver are released down a duct into the small intestine as well. So we want it to go nice and slowly because we want it to mix with all these substances from the liver and from the pancreas, the enzymes from the pancreas. So we'll talk about the liver in quite some detail later on. So the pancreas is a endocrine organ. It's also an exocrine organ. So that means it's releasing hormones. Endocrine and exocrine just means it's either releasing stuff down a tube that we call a duct, or it's releasing stuff directly into the blood, and it does both. But for the purposes of what we're talking about now, digestion, it releases stuff down a tube, which we call a duct in biology. So we've got this chyme, this mixed up food and gastric juices from the stomach being released a very little amount at a time into the small intestine. We've got enzymes being released down this duct called the pancreatic duct from the pancreas. And from the liver, we've got substances that have been produced in the liver and then stored in the gallbladder and then released down another duct called the bile duct, just another tube into the small intestine. So there's a lot going on. The main stuff from the stomach, and then we've got these accessory organs, the liver, the gallbladder, the pancreas, all releasing stuff in to mix with the food. So the main part of digestion of our food actually happens in the small intestine, even though we've been talking a lot about it starts in the mouth, it continues in the stomach. The main bit, most of digestion and the terminal digestion takes place here because of all of these enzymes from the pancreas. There's many enzymes secreted down this pancreatic duct. We have got something called trypsin and chymotrypsin, which is protease enzymes, so further breakdown of the proteins into these amino acid subunits. So you just get an individual amino acid, which can get easily moved into the blood. And then we've got pancreatic amylase, which is a starch digesting carbohydrate to further digest any starch. We've also got maltase that we were just talking about, which breaks these disaccharides down into glucose. And we've got lipase. So this is the fat and oil digesting enzyme. So lipase breaks down lipids. So this is what a lipid looks like. We've got 3 hydrocarbon chains. Got a blue one, a green one, and a red one in this picture. That just means it's a chain of hydrogens and carbons. And then we've got them attached to this little black molecule here, glycerol. And the lipase enzyme will split that up. So that breaks it down, not into subunits, because the protein was in a big subunit, amino acid, amino acid, amino acid stuck together. The carbohydrate had loads of subunits, glucose, glucose, glucose. The lipase is the lipid, sorry, which is broken down by lipase, is a little bit different. We've got these long hydrocarbon chains stuck to a backbone molecule called glycerol. So we've got proteases digesting proteins into amino acids. We've got amylases digesting starch, like maltase is there as well. And in addition to all of this, we've got bicarbonate, which is being released as well. What this does is it neutralizes that acid from the stomach. Remember we said that chyme had a pH of about 1 or 2, which is really acidic. And we get this bicarbonate released from the pancreas along with all of these enzymes. And this has got a much higher pH. So it's got an alkaline pH of around about 8, 9, 10 maybe. And that will help to neutralize this very acidic stomach content, which is really important. Remember, the stomach was protected by this mucosal layer, and the small intestine doesn't have as much protection. So we need to neutralize that stomach content pretty quickly. Just check the chat. You can see someone's written something there. So is that why some people drink bicarbonate of soda when they have reflux? Yeah, because that reflux is acid stomach content escaping into the esophagus. Essentially, you've got a sphincter muscle at the top of the stomach. You know, I said the contents from the stomach are emptied into the small intestine a very small amount at a time. There's this muscle at the bottom, a sphincter muscle. That just means a circular muscle that stops it all going whoosh. We have another circular muscle at the top, another sphincter muscle to stop it coming back up. I mean, it can come up. We can vomit. Some animals can't. So we can get it up, like particularly if we've got food poisoning and we need to get that content out, we can get it up. But most of the time we have it contained within our stomach. Occasionally we get acid reflux into the esophagus, and obviously it will burn because it's got a very low pH. So a lot of people take bicarbonate to help to neutralize that that acid reflux. It's very common during pregnancy, actually, when there's a lot of pressure, obviously, on the internal organs, because you've got a whole fetus developing there. So that's a lot of the stuff that's coming out of the pancreas, but also we have contents coming from the liver. So we have something called bile, which is produced in the liver. We will talk about that later on. It's produced in the liver. It's stored then in the gallbladder, and it is released then into the small intestine down the bile duct. Remember, duct is just a tube. So the bile duct is literally just the tube coming from the gallbladder. So bile produced in the liver, stored in the gallbladder, released down the bile duct into the small intestine. The bile is also very alkaline, around about pH 8. Well, it's not very alkaline, it's a little bit alkaline. It's around about pH 8, which will also help to neutralize this chyme from the stomach. So we've got the bile, alkaline. We've got the bicarbonate from the pancreas, all helping to neutralize this very acidic chyme. And the enzymes which are released from the pancreas, you know, the ones we were just talking about, the trypsin, the chymotrypsin, we've got maltase, which is present in the small intestine as well, and pancreatic amylase. All of these enzymes work best in slightly alkaline conditions because that's the pH of that area. And these enzymes then catalyze most of the terminal steps in digestion. So we've gone from ingesting proteins to having amino acids in our small intestine. We've ingested big complex carbohydrates like starch. We have glucose in our small intestine. We ingest lipids, and we've got fatty acid chains and glycerol in our small intestine. So what is this bile doing then? We just talked about it being produced in the liver and stored in the gallbladder and released. It's not an enzyme, but what it does is it emulsifies the fats and oils. And what we mean by emulsifying is we increase the surface area of fats. So when we eat our food, we have these fats, and fats will, because they're in a watery environment, because obviously everything that we eat is sort of plant-based and water-based, these fats and oils will coalesce into big blobs of fat. Now, that's okay, but the enzymes that we then need to break down these lipids will only be able to get to the outside bit, to the surface bit of these big blobs. So that's a pretty slow process, because you imagine there's quite a big volume of fat in here, in the middle of the fat blob that we need to break down. So what the bile does is it breaks up these fat blobs into much, much smaller droplets like these ones here. So it's still have the same volume. It's not getting rid of the fat in any way, but it's just breaking them up into much, much, much smaller droplets, which can massively increase the surface area for the lipase enzymes to work on. So we've got this process going on called emulsification, which increases the surface area of all of the lipids that we've eaten into much smaller droplets, so the lipase enzymes can work to digest them. Find it crazy how all this starts and forms from a zygote. Yeah, it's a little bit mind-blowing. If you think that's crazy, wait until you start to look at some of the molecular mechanisms, such as protein synthesis. That's crazy. So we've got this huge volume of fat, surface area increased, lipase enzymes able to work on it and break down those lipid molecules into fatty acids and glycerol, which we can then absorb. Just take time. Okay, I'll just go through the absorption then. So what we also have in the small intestine are specialist structures called villi and microvilli, and they are adapted for the absorption of our food. And one of the main adaptations is they have a huge surface area. So we're in this bit here, this long wiggly tube. To start with, that tube is really, really long and it's really wiggly. So you can pack a huge length of tube into the area there. Now, if you looked at a cross section of this tube, this light bit here is the lumen. Remember, that means the hole through the middle of a tube. And that's incredibly folded. So it's not a smooth circle. There's a huge folding of the surface of the lumen. So on these folds, there are small folds themselves. These are the actual finger-like projections we were looking at. Remember, we were looking at these adaptations last week when we were looking at specialist areas of, I think, epithelial tissue. Yeah, it was epithelial tissue. So we have these finger-like projections called villi. And if we zoom in onto the surface of the villi, the cells themselves have finger-like projections called microvilli. So these are specialist epithelial cells with microvilli. These weren't for secretion like the ones we looked at last week, but these are for absorption. But still, it massively increases the surface area like the ones we looked at last week. So the small intestine itself is very, very long. It's very highly folded. That means we can fit loads of small intestine into that space. The walls of the lumen themselves are also highly folded, massively increasing the surface area. These folds are covered in finger-like projections called villi, massively increasing the surface area. And on the surface of the villi, we have microvilli, which look like almost like hairs. They're so tiny. These microscopic structures called microvilli, which is an epithelial cell adaptation, hugely increasing the surface area. So another thing that's an adaptation of the microvilli is they are 1 cell thick. So you can see, look, 1 cell there between the lumen of the gut. So this is the bit of the tube that the food goes through here, and the blood vessels here. So there's only 1 cell. So there's a really short diffusion distance for the food particles, so the amino acids, the glucose, to travel. So we've got a large surface area, we've got a short diffusion distance, and then inside these cells, we have capillaries. So that maintains the concentration gradient. So there's always a gradient where there is a high concentration of glucose here compared to a lower concentration in the blood. It's kept low because that blood keeps moving on, because our blood pumps around our bodies. So as soon as we move glucose across from the gut into the blood, that blood moves away and transports it. So we maintain this concentration gradient, which helps us to move stuff. So that's another adaptation. So we've got a large surface area with all of these structural adaptations, 1 cell thick, so that's a short diffusion distance, and we maintain this concentration gradient with our continually pumping blood. So that's the blood vessels, the capillaries there that gives us that. Okay, let's have a little look at some questions then, just before it's almost time to go, and then I'll give you some time to ask me some questions. And we can carry on from this next session. So where is the bile produced? What's the primary function of the stomach? And which section of the digestive system is responsible for nutrient absorption? So we were just talking about that one. So just try it in rough if you don't want to write in the chat, that's absolutely fine, but go through and try and answer them. Okay, I'll just pop the answers up for you. So bile is produced in the liver, is stored in the gallbladder, and then from there it's secreted down the bile duct into the small intestine, where it emulsifies the lipids, so spreads them out and increases the surface area. The primary function of the stomach is to store the food until it gets passed on into the small intestine. Remember, it's just a little bit at a time is released so that the enzymes have got time to break down the food into the smaller, more soluble, absorbable subunits. And obviously it mechanically and chemically actually digests the food. So it churns up the food, this thick, strong 3 layers of muscles churning up the food, further mechanically digesting it and mixing it with these protease, enzymes, pepsin. And the section of the digestive system that's primarily responsible for nutrient absorption is the small intestine. We're just looking at the adaptations there, the large surface area, the good blood supply, the short diffusion distance, so short distance for the molecules to move across to the blood. I think what I'll do is stop on that slide and we can start back up here on the next session, and that will help us to remember what we were doing a bit. So I think that would be a good place to stop. So have you got any questions about any of this stuff for me before we go? If not, I can take any questions you've got on Saturday if anything comes up, and I will see you next session. Do the intestine cells produce mucins? Yeah, there's mucus produced exactly for the purpose there of protecting that in the stomach, not the intestine so much. There'll be a little bit, but they're not anywhere near as protective as the stomach. So most of that small intestine protection is to do with neutralizing the contents of the stomach. There's a lot of enzymes in the wall of those cells in the small intestine. So we need substances to get across that small intestine because it's absorption of food molecules, and there's actually enzymes embedded in the wall of the structure. So there's obviously not cell walls, they're animal cells, but there's a lot of enzymes embedded in the walls of the structures there. So we need the substances to be able to get to them. So it changes along the structure. Remember, it's got those three sections, the duodenum, the ileum, etc. So the structure does change a bit as you go along the length of the tube. But yeah, there's different adaptations as you go along. Okay, so I will see you on Saturday, and we'll carry on with this Saturday then, or you can watch the recording, obviously, if you're not available. Bye, guys. Bye-bye.